and welcome to week 2 of Embryology. I am Tanya Chamberlain from the Division of Anatomy here at the University of Leeds, and in this video we are going to be looking at the full implantation and the formation of the bilaminar disc. After this lecture and completing any further necessary study, you should be able to describe the differentiation of the trophoblasts, describe the differentiation of the embryoblasts, and appreciate the importance of these embryoblasts, differentiations, epiblast and hyperblast, into establishing a dorsal ventral axis of the embryo. You should be able to describe the process of full implantation of the blastocyst and the formation of the amniotic cavity and the yolk sac, and describe the production of HCG and its importance in pregnancy testing. You need to be able to discuss the normal and abnormal implantation sites and the consequences of these abnormal implantations, and describe the formation of a hydatid E-form mole. And this is the way the lecture is going to plan out. So first of all, we're going to look at trophoblast differentiation, followed by embryoblast, which is establishing the dorsal ventral axis. And then we're going to look at the implantation of the blastocyst, and then the role of HCG, the abnormal implantation sites, and the hydatidiform moles. So we ended our journey last week on the fertilized zygote with its cleavage until it becomes a morula and then eventually a blastocyst. And we know that these cells within this blastocyst are going to continue to divide and become more organized and more specialized. We know that we have an inner cell mass, which is formed of embryoblast cells, and we have a outer cell mass, which is formed from these trophoblast cells. And these trophoblast cells are what we're going to look at first. Trophoblasts are going to form the crust, and the inner cells are going to go on and form the embryo. So these outer cell blasts are going to differentiate into cytotrophoblasts, and these are going to keep a very structured boundary to their cells. And then we have these syncytiotrophoblasts, and these are going to lose their cell boundaries, so their membranes are going to disappear. And these are going to form a syncytium, which is essentially a fluidy pool containing lots of nuclei. And these syncytiotrophoblasts are going to produce proteins to modify the maternal immune system and prevent rejection. And because they don't have much in the way of membranes, this is going to prevent identification of stray proteins and prevent the maternal immune system reaching the epiblast, because of course all of these cells contain 50% foreign DNA. These contain 50% paternal DNA, which the mother's immune system, the maternal immune system, is not going to like. So these inner cell mass here we have in yellow and blue are going to form are going to differentiate from these embryoblast cells into hyperblast and epiblast. And we have the hyperblast cells here in yellow and the epiblast cells next to the endometrial lining and form epiblasts. So there is a significance in becoming bilaminar, in that it is the first stage of organization that we really get into. So this is going to form where we have all of our organs essentially, and this helps to establish this dorsal ventral axis of the embryo. So we can see on the dorsal aspect we have these embryoblasts forming, and on the ventral aspect we have these hyperblasts forming. And these essentially are what mean we kind of reverse into the endometrial lining. So we are reversing in epiblast cells first into the endometrial lining, and the hyperblast cells remaining ventral. And then we have a formation of the amniotic cavity. So within these epiblast cells, a space is formed, and this space is going to become the amniotic cavity, and this is going to grow as more and more of these epiblast cells continue to differentiate and reproduce, so replicate, this cavity is going to get larger, and we will look at that in a few moments. We can also see that these syncytial trophoblasts are beginning to impenetrate the endometrial lining, and eventually they are going to reach this maternal capillary. We can see that these cell membranes of these syncytial trophoblasts are beginning to disappear. So we have less of a defined boundary. So now we are beginning to have what we call the full implantation. So the syncytial trophoblasts are continuing to move through the endometrium of the uterus. And they're going to reach the uterine glands and the capillaries. And as they do this, they are going to form what we call trophoblastic lacuna. So these trophoblastic lacuna we can see here, and these are spacers, which essentially the proteins from the syncytial trophoblasts are causing the maternal capillaries to become leaky, and this is leaking blood into these lacuna, and these are forming the beginnings of a utero-placental circulation. So within this blood we're having diffused oxygen and nutrients that's going to supply these embryoblast cells, so these epiblasts and these hyperblast cells. The hyperblast cells are also beginning to migrate, and these are migrating around the ring or around the perimeter of the blastocyst cavity. And as they are doing that, they are going to go on and form what we call a Hos's membrane. And this Hos's membrane is going to encase itself around the blastocyte cavity, and this is going to form the primary yolk sac. We also have the development of extra embryonic mesoderm tissue, and this is going to form around the outside of that primary yolk sac, so external to Hos's membrane. And this extra embryonic mesoderm is going to go on and form the placenta and the umbilical cord. We have further erosion of the maternal capillaries by these syncytial trophoblasts, which are going to allow for blood to pool, and these lacuna are getting larger and larger, filled with maternal blood, which is going to go on and supply the embryo tissues, so the epiblast and the hyperblast cells. By days 11 to 12, we can see that this extra embryonic tissue has completely encircled the whole of the epiblast and the primitive yolk sac. We can also see that the amniotic cavity is continuing to increase in size, and the blood being supplied by these lacuna from the syncytial trophoblasts is going to continue to increase as utero-placental circulation is becoming more specialised. So then we can see as we reach days 12 to 13, so this has all happened in a very small amount of time, that the extra embryonic mesoderm has completely encased the what is the primitive embryo, and we have formed a chorionic cavity within. So we have some separation of this extra embryonic mesoderm, and the chorionic cavity has formed. During the days 12 to 13, we have more hyperblast migrations, and as we can see here, these hyperblast cells are continuing to migrate, and these are going to form the secondary or the definitive yolk sac. And as they continue over the next couple of days, the next 24 hours to 48 hours, these hyperblast cells are going to continue to migrate more and more, and as they are doing so, they are leaving essentially what is the rest of the inner cell mass. So they are removing themselves from the inner cell mass, and they are going to form a secondary or definitive yolk sac, and the remnants are going to be expelled towards the outside of the chorionic cavity. So this chorionic cavity is getting larger and larger, and we have the definitive yolk sac, which is formed by this secondary migration of hyperblasts, and the remnants of the primary yolk sac are pushed to the wall to within the rest of the extra embryonic mesoderm. And this is how it's going to look between the days 14 and 15. So now we have what we call full implantation. So full implantation has arrived, and we can see that we have these syncytial trophoblasts around the outside, which has formed these huge trophoblastic lacunas now, so these huge lakes of maternal blood that are now fueling the oxygen and nutrient requirements for this embryonic tissue. We also have a nice defined boundary of the cytotrophoblasts, and we have this whole encapsulated area of extra embryonic mesoderm, and we have the development of this connecting stalk. So this is formed by this extra embryonic mesoderm, and this is going to go on eventually and produce the umbilical cord. Now, something that we rely on for detecting pregnancy is the presence of HCG, so human chorionic gonadotrophin. And this is the hormone that's released, and this is released by syncytial trophoblasts. So in addition to its role as an immune hider and a blood supply creator, these syncytial trophoblasts are also very busy in that they are producing or they are signaling for the production of human chorionic gonadotrophin. And this is used to maintain the endometrium and play the role in maternal immune tolerance. So again, this is going to help dampen the maternal immune system and continue to hide the pregnancy from an immune response. And we test this by using urine test strips. So this is a very simple and non-invasive way of testing for pregnancy, in which the maternal figure urinates into either a cup or urinates directly on the stick. And once the threshold of HCG is met, that test will show up positive. These tests are becoming more and more accurate, and so are requiring a lower threshold, and therefore we are able to test before or sooner throughout pregnancy, which is particularly important when certain criteria have to be met. For instance, if the maternal figure is likely to have a complicated pregnancy through to comorbidities. So let's have a look at some implantation sites. So the uterine tube cannot support pregnancy, and given that we know that fertilization often happens within the ampulla, which arguably is a little bit further along in this image. This I would describe as being the infundibulum, but hey ho. Given that the fertilization normally occurs within this region, occasionally we can have implantation of the blastocyst into the ampulla, and of course this is not designed to support pregnancy. So apart from its sort of narrow diameter, it also has less of a blood supply than the uterine cavity because of the thickness of the endometrium. And therefore, if we have an implantation site, most of them will spontaneously abort, but it may be a case of that this is going to become a medical emergency and have to be managed with either surgery or abortion. Sometimes we can also have that the fertilization has happened in this space between the ovary here and where the fimbriae will collect that oocytes, that mature oocyte that is ready for fertilizing. In the event that this happens, we can actually have migration of a fertilized zygote, and it can implant onto the mesentery of the intestines. So this can become an extrauterine pregnancy or an abdominal pregnancy. And again, this area on the whole is not conducive to supporting a developing embryo or developing fetus, and there can be a lot of complications that arise from this, including both fetal and maternal death. In very rare instances, we can have an ovary that is ending up being the implantation site for a fertilized oocyte. So a zygote can, or a blastocyst rather, can try and implant on the ovary. Or we can have what is technically not considered an ectopic pregnancy, but a very inconveniently placed implantation site, and that would be over the internal os of the uterus. And this can lead to a condition called placenta previa, which can have high rates of maternal and fetal death. And so, typically speaking, this would be managed by monitoring throughout growth, so with the use of ultrasound scans, and probably, unless the placenta is able to move away from the uterus, away from the internal os, would likely result in a caesarean section. And this is what we can see here. So we could see that this was probably a cervical or an internal os implantation, and so the placenta has now formed over the cervix, which, when you have a 7-pound bundle of joy bouncing on a very highly vascularized organ, you are increasing the risk of premature rupture of the placenta, so the premature detachment of the placenta, and essentially hemorrhage for both maternal and fetal parties. Also, during labour, obviously the cervix, the internal and external os, are going to move apart during contractions, and this has the potential to rupture those vessels, this capillary system within the placenta, and again increases the risk of maternal and fetal mortality. And here we have an image of a tubal implantation. So this is where the majority of ectopic pregnancies occur, due to the location that we would expect the mature oocyte to become fertilized. And if we have an abdominal pregnancy, we often find that it is in this retrouterine pouch here, largely because it's the closest, and it also contains, which we know, a lot of peritoneum, and then we have the mesenteries for the intestines as well, located in this region, which are also highly vascularized and can typically support pregnancy for longer. There has been a couple of documented cases where an abdominal pregnancy has survived almost to term, and delivered with maintaining survival for both of the parties involved here. Now, in the event that these ectopic pregnancies develop, or these sort of in the event that abdominal pregnancy in the event of an abdominal pregnancy, quite often the fetus or the embryo will self-abort. But if it grows too large, it cannot be reabsorbed, and obviously, given where its location is, it cannot be passed out through a cervix because it is not inside the uterus. So what the maternal body tends to do is it calcifies these remains and forms essentially what we know as to be a stone baby or a lithopedion. Now we also have a hydatidiform mole, and these are the development of the trophoblast without any embryonic tissue. So this is essentially there are no epiblasts formed and there are no hyperblasts formed. So this is just basically an empty egg. And what has happened is the oocyte didn't actually contain a nucleus, and so fertilization of an empty cell has formed the outer, so the kind of the extra embryonic cells. So these are the trophoblasts, but it hasn't formed an embryo itself. And this is what suggests that paternal genes favour the formation of these trophoblast cells. And the reason that we suggest, the reason that we can come up with that offer explanation for this is because it's in the paternal figure's interest to hide their cells from, to hide the embryo from the maternal immune system. So we have these syncytial trophoblasts that are produced to immediately hide the fertilization of, or the fertilized product of conception. And because these syncytial trophoblasts produce HCG, this can come up as a positive pregnancy test. And it's often not until later within the pregnancy, when a maternal figure goes and has an ultrasound scan or experiences a spotting and goes for an early pregnancy scan, that this kind of anomaly is found to exist. So what can often be described as it was an empty sac, and that is essentially what a hydatidiform mole is it is a sac without an embryo within it. And that is the end of this video. In the next video we will move on to look at the process of the differentiation of the embryoblast cells, so those epiblasts and those hyperblast cells, and how they are going to continue to form the blueprint of the anatomy we are interested in.